Let's move to the last talk of this meeting by Miguel Calero, who's a researcher at the Instituto de Salud Carlos III, and he works on the molecular characterization of prion diseases. And the name of his talk is already on the screen. So that's.
Thanks, Isabel. Thank you to the organizers for the invitation to this meeting. And thanks to all of you that still remains here for the last talk of the meeting and after 3 long days. Well, our group at the Instituto de Salud Carlos III is a heterogeneous group composed of 3 subgroups. The first one, led by Dr. Jesus de Pedro, is mainly on research on public health and aging. The second subgroup is led by Dr. Pablo Martinez Martin, and their research is on clinical neuroepidemiology and results. And my group is focused on molecular biology in neurodegeneration. Besides that, we are also part of the working on the registry and surveillance of prion diseases and on clinical diagnosis of prion diseases. When we were asked to give this speech, we thought, because we work in different neurodegenerative diseases, to give an integrated model of neurodegeneration, starting from the molecular basis of the disease and giving some hints of the epidemiological and clinical implication of that model. This part of the data and facts that we discuss in the presentation are already published in these 3 papers. And the basis of the model that we want to discuss today is based on the conformational basis of the transmission of these neurodegenerative disorders in the central nervous system. This concept has been already coined in the concept of molecular nexopathies. These molecular nexopathies are based mainly on protein misfolding and conformational conversion, transcellular disease propagation through brain networks, and network disintegration. In order to better understand what conformational diseases are, we are going to give some brief hints of what is molecular biology of these problems. Here in this table, you have some very well-known problems such as Alzheimer's disease, prion diseases, tauopathies, Parkinson's disease. In all of them, we have a protein that get deposited, and in many cases, that deposit is a pathological hallmark of the disease, as you know. Many of them, as you see here, are pathologies exclusively of the brain, perhaps because the brain is the most vulnerable organ of the body. From all of these disorders caused by the deposition of a protein, this protein, as you see here, are very different and has different functions. Therefore, nor primary sequence nor function are key features of these conformational disorders. Rather, protein instability or tendency to misfold and susceptibility to conformational mimicry are the core of these disorders. In fact, at the very core of these disorders is the fact that one single molecule, one protein, is able to adopt at least 2 different conformations. One that is the normal conformation, which is doing its role in the cell or in the body, and another protein, which is the pathogenic protein we call the incorrectly fold, and this protein, the incorrectly fold protein, is able to catalyze the conversion of the normal protein to the pathogenic protein. In most cases, this conversion is associated with an increase of beta sheet structures, decreased solubility, a higher tendency to aggregate, and higher tendency to resistant to be degraded by proteases. These conformational changes are associated to both protein loss of function. The protein does not the function that it was doing with the normal conformation, and also gain of function, usually toxic gain of a toxic function. Based on this model, molecular transmissibility of these proteins is based on the propagation of the abnormal conformation to a constitutively expressed protein. Thus, it requires that at the same time, simultaneously, we have the presence of the abnormal isoform and the de novo synthesis of the normal protein. This is, at the molecular level, one of the most accepted models of conformational conversion, and it's called the nucleate polymerization. This model assumes that both the normal protein and the misfolded protein are coexist in an equilibrium that is very displaced towards the normal protein. This is in an energy diagram is shown by the protein. The normal protein has a lower energy, so it's more stable than the misfold protein. So this equilibrium is to the left. But when this protein, the abnormal protein, aggregates, it gets a lower energy, is more stable. And this makes that the equilibrium goes to the right, producing more and more abnormally folded protein. This simple model may explain the dual etiology of most of the neurodegenerative disorders, genetic and sporadic, and even in some cases, such as prions, even the infections origin of the disease. In this case, genetic cases will produce abnormal proteins, mutated proteins that are produce either more stable abnormally folded proteins or less stable normal proteins, inducing this equilibrium go to the right and facilitating the conformational change. This model also has some important implications for the diagnosis of these disorders. In our view, it's important not to detect these conformers aggregated, such as in the plaques or in the tangles, but it's important to detect the conformers that are normally folded at that propagate the conformation. Also, for this model has therapeutical implications that just looking at this diagram, we can think of different approaches to prevent the production of this misfolded protein. For example, by decreasing the synthesis of the normal protein or upgrading or upregulating the degradation of the protein, either the normal or the abnormally folded protein, or facilitating the clearance of the aggregates. But the disease is not produced by the conformational change by itself locally. The disease is produced by the propagation of the conformation to different parts of the system. And in this case, of the different parts of the brain. Just 2 days ago, Professor Del Rio explained to us the main five mechanisms by which misfolded proteins can go from one cell to the other. Namely exocytosis, exosome transport, tunneling nanotubes, receptor-mediated transport, and endocytosis. This is key for these disorders. If we don't have cellular transmission from one cell to the other, we will not have disease. Once we have a mechanism of transmission of the misfolded protein from one cell to the other, these misfolding proteins propagate through the brain through specific brain networks. This is what is called molecular nexopathies, which are defined as specific coherent conjunctions of pathogenic proteins and intrinsic network characteristics that define network signatures of the different neurodegenerative disorders. And this concept has three key features that are differential intrinsic network vulnerability, differential vulnerability of neuronal connections to particular pathogenic proteins, differential impact on molecular effects. These vulnerability patterns are, of course, determined in part by genetic background, and it shows these vulnerability patterns both at the protein level. Different proteins has different attack different vulnerability pathways at the cellular level, also at the anatomical level. These vulnerability patterns define the different clinical phenotypes that at the end results in different neurodegenerative disorders. What is the vulnerability pattern? In this slide, we show has different proteins has some specific pattern of vulnerability, which the disease, the protein, the misfolded protein is spreading through the brain. Each pattern is different for each protein, and that different patterns give us the conjunction of the proteins and the pattern give us at the end different disorders. One example of this specific pattern of dissemination is a paradigmatic example is the case of the protein, the prion disorders. In this case, the same pathogen, the prion protein. It spreads in different patterns according to different vulnerability, producing different spectrums of the disorder, namely Creutzfeldt-Jakob disease, Kuru, GSS, fatal familial insomnia, or variant CJD, which affect to different regions of the brain, let's say the cerebellum, hypothalamus, or cortex. And an extreme example of these differences in vulnerability patterns is the case of the mutation at codon 179. This is a mutation associated to human prion diseases, and this mutation, D4N, depending on the polymorphisms at codon 129, will determine that we have either a phenotype of Creutzfeldt-Jakob disease or fatal familial insomnia with very different regions affected by the same pathogen with a small change in one polymorphic amino acid. This model also explains the phenotypic conversion that is seen in many neurodegenerative disorders. That's why we call them mainly, we have two very broad groups of dementia, of movement disorders. Even that the disease is starting in a different network, the disease as progresses, it ends affecting many systems that resembles one entity to the other. In this slide is shown that as different pathologies, as Alzheimer's, frontotemporal dementia, ALS, or Parkinsonism, has different proteins involved in the disorder, but at the end, these disorders affect different areas that at the end show similar clinical phenotype, showing a clear overlap among conditions. In this case, this is you can see here how the pathology in case of A-beta or tau in AD spreads through the vein, alpha-synuclein in Parkinson's disease or TDP-43 in the case of ALS. Now coming from the individual, from the brain to the population and looking for implication in public health, in this diagram, we are seeing the incidence curves from different conformational disorders. This is a very interesting graph, and done mainly by Dr. Jesus de Pedro. In this graph, you can see this is a normalized incidence of neurodegenerative disorders. So even though that this disorder has very different incidence, what we are seeing here is that the maximum incidence is normalized to 1 for each disorder. So at the beginning of the graph, we can see those that are acquired, such as variant CJD. In the middle part of the graph, we have some genetics forms, such as genetic CJD or genetic cases of Alzheimer's disease. And at the end, at the end of the graph, with the incidence at the end of the life, we have the most prevalent disorders such as Alzheimer's, Parkinson, etc., or Lewy body dementia. From these graphs, some important findings are relevant. That is that those, as you can see here, those disorders that appears early in life usually have a more aggressive phenotype, evolves very rapidly, and has a very low incidence. But those that appears at the end of the life has an incidence much higher and also a longer duration of disease. This and some other findings has led us to look for drivers that are common factors to different these neurodegenerative disorders. Agustin just mentioned that some genetic factors are common to several disorders, but I don't have the time to discuss in detail all of these drivers, but I would like to mention the two first that are very epidemiological related. We have already mentioned the relationship between the second driver, relationship between age at onset, survival, and incidence, but also the first one is very interesting from an epidemiological point of view. Coming from, as I mentioned at the beginning, we are coming, the group, we work on prion diseases, and in these disorders, there is clear pattern that some disorders are specifically, we have a specific age-dependent susceptibility to contract, to get the disease. For example, in variant CJD, only persons that are young were infected by the mad cow disease. In some other neurodegenerative disorders, we can thought of similar processes, but the idea is that we have specific patterns of susceptibility that are very dependent of the age of the exposure. So besides taking into account environmental or viral factors or whatever, we should have into account when that factor affects to the person. In this model, we just try to gather the information, several risk factors and drivers, and to try to explain how these different factors modulate the different neurodegenerative disorders. One example of the implication of this approach is the proposal that we have done for the decrease of transmission of Creutzfeldt-Jakob disease from a previous paper from our group. Dr. De Pedro's group demonstrated that there is a transmission by surgical procedures, but that transmission is based on surgical procedures that were done 20 years before the onset of the disease. And also, we know that the people who develop these disorders, sporadic so-called disorders, are persons that are all 62 years old mainly. So if we tried to minimize the transmission of these disorders with that long incubation period that affects mainly to all people, we can propose, having in mind all that data that we already know, that if we, this is in a surgical procedures, this is a person which is operated, and then some other person is operated afterward. So if potentially some transmission could occur from this person to this other, if we operate on this person, persons on these ages, these persons are more likely to have a disease of this type, and could, if we transmit this disease to persons that are more susceptible in this range of ages, there is high chances that they will develop the disorder. But opposite to this, if we operate first on persons that has lower risk of carrying the disorder, and afterwards we operate on persons that are older, there is less chances of transmitting the disease, and even if this person got the disease, will not develop it because the incubation times are very long. So just to finish, because I'm running a little bit late, just I would like to mention of this kind of conclusions, that this common approach may allow us to improve our techniques for the diagnosis of these of these disorders in general. We are on the verge of having more sensitive techniques such as single molecule counting, or techniques of amplification of misfolded proteins such as PMCA or RT-QuIC. These techniques may allow us in the near future to detect the pathogen that is really transmitting from cell to cell, rather than the plaques or the tangles or any other proxy of the disease. Also, we think it's very important, as Agustin mentioned before, to understand the interaction of this model with comorbidities such as vascular, diabetes, etc. Just to finish, this is our group at the Instituto de Salud Carlos III, and also I would like to mention to Dr. Alberto Rábano, who collaborated in all the papers I mentioned before. Thank you very much.